unSILOed podcast is produced by University FM, elevating the stories of your institution.
Welcome to unSILOed. This is Greg LeBlanc, and we're here today with Laurence Hurst, who is a professor of evolutionary genetics at the Milner Centre at the University of Bath in the UK, and also the author of this book called The Evolution of Imperfection, the science of why we aren't and can't be perfect. Welcome, Laurence.
Thank you. Glad to be here.
Well, look, I like to see similarities between evolutionary biology and economics, right? Because in economics, we're always interested in, right, why we, we can't achieve this hypothetical model of efficiency. And I think in biology, I mean, I don't know how common it is for people to talk about sort of perfection, right? And imperfection. It's kind of an odd thing to talk about.
Perfection is quite common, yeah. And I think you can understand why, in so much as actually the evolutionary process, and it is similar to economics in many regards, but the evolutionary process, if you take the simple narrative that maybe we've all heard of the evolutionary process, survival of the fittest, and that's really nicely captured in, the march of progress. So if you take the word evolution and you put it into Google image search, just about everything that comes back is the same thing. It's this knuckle walking chimp gradually going upright. And of course, there's the last figure is often a little bit of a joke about, how we're de-evolving or whatever. But the underlying assumption is things are getting better and better and better. Humans are the top of the Christmas tree, as it were. And then if you think about how evolution works, we would often tell you the story of the pepper moth, where we got very good evidence for natural selection. And so in the 19th century, this is how the story goes. In the 19th century, in northern England, the trees went black because they got covered in soot during the Industrial Revolution. And there were two sorts of pepper moth within the same species. One was a black moth, one was a white moth. And the black one then rises up in frequency because it's eaten less, because it's camouflaged against the black background. And then latter part of the 20th century, the Clean Air Act was introduced and the black trees went to be white, and then the white version comes up in frequency. And so what you get is the idea that the evolutionary process is really rather straightforward, that the better ones outcompete the less good ones. And if you just iterate that process over and over, you get perfect camouflage, for example. And if you look at something like camouflage, you get a lot of that camouflage is utterly amazing. It is astonishingly good. And so, yes, the Darwinian adaptation, a priori, unless you put something else into the mix, should lead to things that are remarkably well adapted to their environment. So yeah, perfection would be the end product of that. And so, yes, in economics, you sort of have the equivalent of let's assume everybody is a rational behavior. But we just assume, let's assume selection is operating. And our assumption, I think, is rather better than your assumption.
Well, look, I mean, there's a whole taxonomy of imperfections, right? There are explanations for imperfections. And we use the same ones in economics. We'll talk about how, the fitness landscape moves. And so the peak is over there, and it's going to take some time for us to get over there, right? So that's kind of
or may not even be able
story.
to get over there. Yeah. So in evolution, one of our classic explanations is encapsulated by the problem of peak jumping. So the idea is you can come to a local fitness peak, but there may be a better solution somewhere over there. But to get there, you have to go down to the valley and back up the other side, as it were. And if evolution can only ever force you up by fitness, then how do you cross the valley, so to speak? So that's one of the classic explanations. And for example, when we look at our eye, has a peculiarity. It's a very good eye. We see very well. But if you look at the back of the eye, it turns out that our light receptors are sort of backwards. And so the nerves come out pointing out into the eye, into the front of the eye. And so they have to be wrapped around and go back again to the brain. That creates a little blind spot. Whereas the octopus eye has exactly the same problem and has solved it the intelligent way, which is also the way we design digital cameras with the information going out of the back, as it were. And there we think is a sort of classic peak jumping problem. You've got a good solution. It may not be the very best solution, but how do you go from having a good solution to a very best solution without de-engineering everything in the process? And that's one of our classic explanations. So we've known about that sort of thing for a while. There are others. I mean, particularly for humans, for example, there's a lot of evidence that we are simply not adapted to the world that we are currently living in. So there's massive rise, for example, in the rates of autoimmune disorders and things like eczema and asthma and hay fever. We see this quite strikingly over the last 50 or so years. And a lot of this is being attached to the fact that it's something to do with modern living, and we're living in a too sterile environment. So, if you want your kid not to get asthma or eczema, for example, just live on a cow farm. And apparently, the bugs that they get are very good for training the immune system. You tend not to get those things. Likewise, our diet is, for the most part, not a healthy diet. And if you look at the rise of type 2 diabetes, for example, that can be very much put at the doorstep of A, medicine allowing us to live longer, but B, our diet, meaning high fat, high salt, high sugar, giving us type 2 diabetes. So what we die of now is type 2 diabetes, cancer, cardiac disorders. These are modern issues because we didn't really ever live in a world in which cancer, cardiac disease, and type 2 diabetes was ever a problem. A, we didn't live that long, and B, our food was rather healthier, we think.
Yeah.
So we don't need that sort of explanation.
Yeah, so path dependence is a big part of the story in economics also, right?
Yeah.
And how you get stuck on these local maxima. But there's also, right, the idea that the genes, the selfish genes, will drive us towards an equilibrium that is not good for the species, right?
Oh, there's no reason anything should evolve to be good, quotes, good for the species. The selection doesn't operate on species, it operates on at the instant, it operates on mutations in populations. And we certainly see cases of some very strange genes, which, for example, there's some in fruit flies where the male produces only half the sperm it should do. All the progeny, it's on the X chromosome, so all the progeny end up being female. And you can run an experiment where this actually spreads very nicely through population, exactly what the theory predicts, and it sends the population extinct. They run out of males. So there's no notion in evolution that things ever operate, quotes, for the good of the species. The only question that's really ever worth asking is, when a mutation is rare, what's going to happen to it? Because mutations come in rare into a population, so that's their period of the highest jeopardy. And then what happens? It may well reduce population fitness. And we have good reason to think that humans are far from being as fit as they might be. We have a very high mutation rate. We've got one of the highest mutation rates going, for example. And most mutations are deleterious most of the time. 5 to 10% of us will have a rare genetic disorder, for example. And we could be better. We could be a lot better. So yes, there's no notion that things operate for the good of the species. One of the classic explanations, though, has to be lag as well. So even if we are in a process of constant improvement, we aren't necessarily there yet. So it turns out, for example, that humans are still polymorphic. So we have different sorts of backs. There's a thing called the node schmall, which can sit on your back. And folks with these have a back that looks much more like the chimpanzee back.
This is where you get slip discs, right?
Yeah. And if you have the chimpanzee type back, because we're now walking upright, these are the people who are particularly prone to slip discs. Some of us don't have those, and it's genetic, and we don't have slip discs. We think that's one of these things that's probably in process. So, give it a couple more million years. We only evolved from chimps, 6 million years ago? In evolutionary time, that's almost an instant. So yeah, we expect things to carry on evolving, but some of the changes that have happened to our environment are so new and so rapid that we haven't had a chance to possibly evolve to them.
Well, what I found interesting about this book is that while imperfection exists in every species, it seems like humans are particularly imperfect, right? Now, is that just because we know so much more about the human genome than we did in the past, that we can find all these things? Because it seems like, we know about yeast, we know about bacteria, right? We know there's a lot of other things we know about, right?
We do know about a lot of other things, but I think the observation is actually rather a good observation. That actually, on many measures, we come out as being really rather poor. So, for example, it turns out that about half of human embryos, fertilized eggs, have the wrong number of chromosomes and they die instantaneously. Not obviously perfect. And you can ask about the same about other mammalian species, and you find that they also have quite high rates. For cows, that rate is about 30%. For pigs, it's about 14 to 18%. For mice, it's about 20%. Humans, we hold the record. So that's one thing. The other is we can now do so-called parent-offspring sequencing. So sequence mom, sequence dad, sequence the kids. Then you do a spot the difference competition. You can work out what the mutation rate is. So we're all born with about somewhere in our 3 billion base pairs of DNA. We're born with about 10 to 100 new mutations. And so you can say, okay, per base pair of DNA, so I said we got about 3 billion of these, but per each one of those, what's the chance it's going to mutate between mum, dad, and making kid? And our number comes in at about 2 times 10 to the minus 8. So that's about 2 in 100 million base pairs you'll find a mutation in. Now, that's very low, and it's low for good reasons. But if you look at all other organisms, they're lower. Even whales are lower than us. But your average insect comes in at about 10 to the minus 9, the average bacteria about 10 to the minus 10 per base pair per generation. And there's one species claimed to be 10 to the minus 11. There's one strange fungus which uses mutation to attack things it doesn't want in its DNA. And with the exception of that one, we've got the highest mutation rate. So there's 2 world records we really don't want. Another one which we appear to be particularly higher is the maternal mortality rate. So the chance that a mother dies in childbirth, ancestrally, not now. Now in America, in the UK, it's about 0.01% is your chance of mortality in childbirth. Still very high, I would say. But ancestrally, wherever we look, both outside of Western healthcare, but by checking birth records more generally, it's about 1% per pregnancy is the chance that a mother will die in childbirth, so it's about a 10% chance of dying during childbirth at the end of your life, as it were. So 1% per childbirth, the chance you die in childbirth is 10% through your life. And you can compare that to, for example, Japanese macaques, so quite a close relative of humans, quite an interesting species as well, because they also have a rather large head given the size of the birth canal. So lots of people think, this is human big head, small birth canals. And they've got the same problem, but their maternal mortality rate is approximately 0. No one's seen it, and yet it's very obvious in humans. There was a very lovely study actually done of Chilean mummies, so not mummies as in, well, actually, yes, but mummies as in females wrapped up and preserved, dating from 3000 BC to about 1000 AD or so. And they found about 100 of these, but over 100 of these, and a good 10% of them showed evidence of having died just in or immediately after childbirth. So childbirth is, for humans, a spectacularly dangerous pursuit. There was a lovely survey done by Forbes magazine, I'm sure you know the folks, of America's most dangerous jobs. And it turns out nothing comes close to childbirth. Childbirth is an order of magnitude more dangerous than America's most dangerous job, which I believe is being a roofer. If you fall off a roof, you don't have a lot of chance of living, I guess. So yes, we hold a number of really rather bad records you really don't want. Highest mutation rate, highest rate of fetal abnormality that causes instantaneous death of the progeny. But we also have very high miscarriage rates as well. So the human miscarriage rates, this is a pregnancy that has established past week 6. And our miscarriage rate is, for a fit, healthy 20-year-old female, it's about 15%. As you go to 40, 45, the miscarriage rate goes up to 65%. That's nearly all very early miscarriage by week 12 or so. These are records no species wants.
Now, of course, as an evolutionary biologist, we want to see all of these things as, features, not bugs, right? We want to come up with some story that says this is the best we can do given the constraints, that there's probably some kind of upside to this. So are these things
not necessarily
evolved for?
Not necessarily. No, I think actually that therein lies the problem, that if you have only one model of evolution the model of evolution which says everything is for the best and the best of all possible worlds. That's Candide, wasn't it, Voltaire? No, Dr. Pangloss, that was Dr. Pangloss, that everything is for the best. And that's sort of how a lot of people think about evolution, that all we have to do is go, well, no, I'm sure it's got some advantage, really, we just don't understand it. Or maybe your model of evolution is wrong. So actually what I would argue, and what I do argue in the book, is actually that simple model is actually wrong. And there's a number of forces which operate to say, actually, we do expect imperfections like that. I mean, let me give you just one example, for example. So a peculiarity of the way that we reproduce is that if a baby dies in utero straight away, the mother actually saves both time and energy. Now, that might seem a little bit obvious. It is actually quite common that a mother didn't even know she had a potential mother, didn't know she actually had a fertilized embryo in her.
And this would not be true for birds, presumably, right?
So now, take the contrast. A contrast would be, let's say, the kiwi. So a female kiwi will invest 25%, a whole quarter of her body mass into the egg. Now, if that is inviable, she's already spent the energy and she still sits on it. So she's wasted time and energy. Whereas in the mammalian case, the embryo simply is dead and the mother just simply goes again often. Or if you're in a multi-brood species, the embryo is dead and the survivors get the resources that would have gone to the one that's now dead. And in fact, in humans, we can estimate the effect of that. It's known as reproductive compensation. You're compensating for the death of one of the embryos. So if you look at twins, for example, twins on the average are about 40% smaller by weight than singletons. So if you had a pair of twins, one dies straight away, the other one will now grow up to the size of a normal singleton. What is the difference in their fitness? So that gives them a 40% boost in their body mass, which from what we can tell from various tables here, there and everywhere, suggests about a 10% increased chance then of surviving through to age 5. And that's a 10% fitness effect. So this is quite a large fitness effect. Yeah, well, that's nice. In many regards, this is a lovely way to reproduce, because if the embryo is dead, mother is saving all the resources and has the potential to reinvest or just simply go again, as it were. So, the period comes and you try again next month to have the baby. And yes, indeed, that is true. But consider this. So suppose there's a mutation and it's recessive, and what it does is kill embryos. And you can ask, how common should this mutation be in the population? So we find mutations always coming into populations like this, killing embryos. And if it's recessive, so that means you have to have 2 copies of the lethal mutation to die, then the only way these are going to be made is when a mother has got the normal version and the lethal version, and dad's got the normal version and the lethal version. And when they get together, a quarter of their progeny then have the lethal pairing, which then die. Those resources then get reallocated. But of course, 2/3 that are having it reallocated to have the lethal mutation. And so it turns out one of the effects of this is that species like mammals actually have more lethal mutations in the population than you'd have expected, because they get this boost every time. So they get a boost in the hidden state, as it were, when there's a good version and the bad, and the lethal version in the same individual. But they get the boost for every embryo that dies. And so the prediction is, and we see some evidence for this, that we should have lots of mutations that kill embryos early, simply because every time that happens, the lethal mutation benefits from it.
Right. But is mutation rate itself a mutation, right? So does the pace with which we mutate increase or decrease based on environmental factors?
Yeah, so this is one of the questions we've been able to get at and really pretty well answer, I would say, over the last 10 or so years. So just to put this into context, the human genome came out in 2003. To make that one human genome cost a little under $3 billion. Took a bit more than a decade to do. We can now fairly routinely do the same for a couple of $100, and you can do it in an afternoon. So these advances in our ability to analyze DNA is utterly transforming my field, because what it means is that we can now go in to any species that we want to and go, well, let's sequence mum, sequence dad, sequence the kids, do spot the difference competition and find the mutation rates, which is a really key evolutionary parameter. And that's how we know, for example, that ours is 2 times 10 to the minus 8, your average fruit fly more like 10 to the minus 9, bacteria 10 to the minus 10. So we see lots of variation that we can now measure. And then the question is why? Why are we seeing this much variation? Well, the first thing we know is that selection nearly all the time says keep the mutation rate as low as possible. As we are sitting here, we have repair enzymes that are basically going repair damage to our DNA. And there's a very good reason to that. And that is that we're fairly well-functioning organisms. All organisms are, by definition, fairly well-functioning. They're the products of selection over time. And, if I were to give you my watch, take it apart and say, could you just randomly make some tweaks to my watch or to the engine of your car or whatever? You're not going to expect to improve the engine of your car, and you're not going to make my watch better.
So the mutation rate could be too high, but it can also be too low.
The mutation rate is always going to be too high. The mutation rate should be 0. If evolution was a perfecting process, the mutation rate should end up as 0, because mutations most of the time are harmful. And because we can't go, I just want to mutate that gene, or I just want to mutate that gene, we don't do that. We can't do that. Mutation just happens. It's just an error. But what can be adjusted is the rates. So we can do experiments, for example, to select for higher rates or select for lower rates in the lab, not for humans, obviously, but for single cell species where you can do this. And you can select for higher and you can select for lower rates of mutation. Then the question, of course, is why doesn't everything have the same and really low rate? So everything's got a really low rate. 10 to the minus 8 is still very, very low. But why isn't ours 10 to the minus 9? Or why isn't the fruit flies 10 to the minus 10 that you see in bacteria or whatever? And we think we know the answer to this one. And it comes out of a body of theory that was first developed by Tomoko Ota in Japan. It's called the nearly neutral theory of evolution. It's one of the 2, I think, grand explanations for why we can't be perfect. And what the theory says in no small part is you have to stop thinking of evolution as this eternally progressive process. And actually, most of what's going on most of the time is mutations are coming in and they're just a little bit bad for you. Most of them will not be lethal. They'll just be a little bit bad for you. And so she asked the question, what happens to mutations that are a little bit bad for you? Well, on the one hand, you've got selection kicking them out of populations. So they come in, selection says, well, you're a bit bad, so that means on the average, you're more likely to be in a body that's going to die or not reproduce. So you're a little bit bad. But opposed to that are just random processes. And so she says, actually, you could think of this really as thinking not like a really deterministic process, but much more like particles of dust floating about in a room. You know that when, the sunlight comes into the room and suddenly you see all these particles of dust. And she said it's much more like that. Particle dust that just float about and they can go up and they can go down. And even though they're a little bit dense, and so on the average, just goes to the floor, you will still find some on top of the cupboard. And you'll still find stuff on the ceiling, and so on and so forth. And you will always find this because they're floating about. And so she says that basically these 2 opposing processes. You've got things floating about because of chance, only chance, and you've got this downward pressure, which is selection. And her basic message is that when you then come and model this, it turns out that, and I think fairly intuitively, that the random process starts to dominate when populations get smaller. And the analogy would be, could you imagine your room has a low ceiling? Well, then the chance of bobbling up and down and actually making it from low down up to the ceiling, that's actually quite good. If you had a very high ceiling, your chance of going up and down and randomly make it to the ceiling is actually rather low, with selection, gravity always pushing you down, as it were. So the core prediction of this theory is that selection is not that efficient a process when populations are small. So this is really a very different way of thinking about evolution, because as I said, most people think evolution, this progressive process, all we have to do is come up with the advantage that this confers. And she says, no, there's no advantage here. The genomes of species that have small populations are going to be much more subject to just this waffling about of mutations, because selection basically hasn't been given enough time You just need a couple of lucky jumps, and then selection is irrelevant, as it were. And so the prediction of this is, it says that things like genome size, genome bloating, should be dependent on population size. And with 3 different measures of genome bloating that are independent of each other, and it's absolutely right. So our genome is nasty and bloated. So one of the great mysteries for our DNA when it came out is only 1% of it is protein coding, and we only have 20,000 protein coding genes. You look in bacteria, so your E. coli that lives in your gut, for example, 85% of its DNA is protein coding, which is the canonical job of DNA. And the rest is control elements for it. So there's a lithe, beautiful genome. And you do the same for yeast. Yeast got a lovely, lithe, beautiful genome. Ours just looks awful. It's just full of rubbish. I mean, so 1% is protein coding, making the genes that we actually need to function, what have you. Another 8% of our DNA is old virus. So so-called endogenous retroviruses. So things like HIV can insert into our DNA. And if they do it in the cells that make sperm or eggs, they can potentially be transmissible, and so on and so forth. So 8% is like old HIV sitting in our genome, just decaying away. And then about 40-50% is old transposable elements, these things that just jump about all over the place for no particularly good reason. And this is a problem of species with small population sizes. Each time one of those things inserts, it has a tiny wee fitness effect. And when the population size is large, it's not going to go anywhere. It's not going to get through to the position where everybody gets it, goes from floor to ceiling, as it were. In small populations, it's got a good chance of going floor to ceiling. So we see that, and guess what the big predictor of the mutation rate is as well? So here we have to slightly tweak the argument. So we say, okay, imagine you had a repair enzyme that slightly increased your mutation rate, so it's slightly harmful. What's its fate going to be in the case of a small population versus a large population? Well, in a small population, it's got a chance of making it by random movement. In a large population, it's never going to get to the ceiling. So they could have exactly the same effect on our quotes fitness, exactly the same probability of us dying. But when the population is large, selection basically is given this long window of time to make sure, no, you're not going to get there. You're not going to get from rare to common. Small population, you have a couple of lucky breaks and you could be there. And it turns out the best predictor then of the mutation rate is simply the population size. And we evolved from a species with our population size coming out of Africa was about 8,000, 8 to 10,000 is the usual estimate. It's tiny, evolutionarily speaking. And so what we're seeing is the hangover. In many regards, it's the hangover of being a large-bodied species with not many individuals in them. So our molecular evolution is largely determined by not make it brilliant to make it perfect. It's largely determined by we can't get rid of the junk. Stuff that accumulates in our garage will accumulate in our garage and loft. The same stuff does not accumulate in the garage and loft of yeast. They get rid of it.
So how do we measure population size then, right? Because, I mean, it seems like we've got a lot of people, right? I mean, is it go back to that bottleneck?
Now there's billions of us. Yeah, now there's 5 billion of us. But historically, what are you looking at when you're looking at me? You're looking at the product of evolution that occurred actually quite a way back, and then the population size was a lot smaller. Now, how can we actually get at that? It turns out there's a very big clue, and that we can get at the population size by actually knowing how different everybody is genetically. So if the population size is really big, the underlying theory says actually there's a large possibility for everybody to be really quite different in their DNA. So and a way to get at that is you can say, okay, let's take any given site and ask what's the chance that your 2 chromosomes are different. Now, if the population as a whole is highly variable, then there's a good chance that your 2 chromosomes are going to be different at any given site. And so we can use that to measure the extent to which we are different, and that then maps directly, if you know the mutation rate, that then maps directly through to the estimation of the population size, the so-called effective population size.
So are there environmental pressures that can increase or decrease the mutation rate?
Well, certainly there will be some for some species, that much we know. So in bacteria, we can alter mutation rates by exposing them to mutagens. And for the cells on our surface, we can absolutely modify the mutation rate by sitting in sunlight, which is why they say use sun cream. What it's actually doing, what that UV is actually doing, is breaking DNA, and if you don't repair it effectively, you've made more mutations. Only mutations in your skin, so you get melanomas. There's not good evidence that it has much of an effect in the germline and the manufacture of sperm or eggs. But as it happens, there are within humans and within mammals more generally, there's some good evidence in mice. There isn't one mutation rate. There are variants of repair enzymes in the human population, which mean that those individuals have a slightly higher mutation rate than the rest of us. Not greatly higher, but a slightly higher one. But yes, you can, to some extent, you can change bacterial environments and they can change mutation rates. So if you really challenge a bacteria, for example, they will do something called the SOS response, and part of that is they increase the mutation rate. But for the most part, the mutation rates aren't particularly plastic things.
Right. So our mutation rate, our high mutation rate, has nothing to do with this hypothesis that humans evolved to survive in uniquely variable environments. I mean, that's the story behind human culture, right?
Yeah, no, there's absolutely no reason to suppose that's the case. There's a series of intellectually difficult problems with that. The first is that if you have a high mutation rate, you die. As I said, we got 5 to 10% of us have genetic diseases, and if you just have a higher mutation rate, what happens is selection simply knocks you out of the population. In fact, you can do the experiment in bacteria. You can actually artificially make bacteria with a high mutation rate. You can delete a couple of their key repair enzymes, for example, and their mutation rate goes up 100-fold. That's quite straightforward to do. You grow these, and what happens is over time they evolve a lower mutation rate. And mutation is not good news. It's what gives us genetic diseases. So that's one problem. There's actually a more subtle problem that is also true in us, and that is that because we're sexual species, if you imagine a mutation that increases the mutation rate, it may make an advantageous mutation somewhere else in the genome, but because of the way we make sex cells, we're randomizing our DNA all the time, and there's no reason that the good effects of that beneficial mutation has an influence on the fate of the modifier that changed the mutation rate. So the modifier that changes the mutation rate sort of sits in a cloud of crap of its own making, cloud of genomic rubbish of its own making, really deleterious stuff, makes that advantageous mutation. Advantageous mutation does very nicely, thank you, but the thing increasing the mutation rate goes down in frequency. Again, you can do that experimentally in bacteria.
Now, the difficulty of reproduction, I mean, is this related to the high mutation rate? I mean, is this a way of kind of getting rid of the defective individuals, so to speak?
No. Again, you're sort of thinking in, could it be some advantage to something? I mean, yeah, it certainly is the case that if you have circulating in a population a mutation that is lethal to embryos, selection can favor it to be lethal earlier. That we know from very nice work done in mice, for example, and that makes perfect sense. So if you're going to die early because it maximizes this thing called reproductive compensation. But a lot that's going on between mothers and infants is nothing to do with things being lovely for everything. So we know, for example, that the placenta, for example, will release products into maternal circulation that we think are trying to manipulate the mother, so increasing the blood pressure, keeping sugar in circulation. So the fetus will release stuff that tries to keep sugar in glucose in the bloodstream. So you get just so-called gestational diabetes when this goes a little bit too far. So the mother's got all this glucose in her blood system. And the mother then responds by making more beta cells in the pancreas to make even more insulin to counter it. And the best interpretation of this was put forward by David Haig at Harvard, and I think it's a good explanation, is that this isn't somehow best for everything. This is actually what we call a conflict of interest. What's best for the embryo is not what's best for mum, and what's best for mum is not what's best for the embryo. So we certainly see things like that.
What's best also for the father, right?
What's best for the father is different again. what's best for the dad's genes in the offspring could actually be to get as many resources out of mother, because if they're not going to be the dad for the next kid, they've got no interest in the survival of the following kids. And in fact, they've got an interest in the following kids not surviving if they know they're not going to be the father. And we see similar things actually in lions. Lions are quite happy to go around doing infanticide, killing off other individuals' babies, because they're not my babies, they're somebody else's babies. That forces the mother to reproduce again, and then has to do it with the fathers who just killed off the babies. So there's a classical conflict of interest again. What's best for dad is not what's best for mum, and it is certainly not what's best for the kids, because they're just dead. And so we think a lot of what's going on in human pregnancies is probably a bit like that. So the blood pressure effects that mothers have can be like that. I don't think preeclampsia is that. Preeclampsia is this condition that affects about 5% going to 10% for older mothers of pregnancies. High blood pressure plus protein in the urine is the usual diagnostic of it. Can kill mothers and babies. The best explanation there is slightly different, and that is that it looks as though the human placenta is particularly invasive. We have a very big brain, and part of having a big brain, we think, is we have to have a particularly invasive placenta. So if you compare our placenta to that of a sheep, for example, so sheep placenta, they're just sort of two things that meet. So placenta, mum, meat, gases exchange. Our placenta does that, really invades the endometrium. So we've got a very invasive placenta, and that's maximizing surface area, and it gives more resources, particularly for brain growth, what have you. But it also has the funny consequence and the unfortunate consequence that if things don't quite work right, that the mother now can see quite well the proteins that have come from the father in the fetus. And of course, that is potentially a disaster. And it's a disaster because, what is the vaccine? We give you a protein and your immune system goes, right, let's attack it. And you have a bacterial infection and your immune system goes, right, let's attack it. And it appears that what preeclampsia is the mother's immune system attacking a fetus because she hasn't recognized it. And there's a couple of occasions where humans really mustn't attack unknown proteins. So we don't want to attack our own gut fauna, our bacteria that live in the gut. That gives you ulcerative colitis or Crohn's disease if you start attacking that. Mothers shouldn't attack the sperm in the vagina because then they'd be infertile. And you don't want to attack your own fetus. So it turns out the big predictor of whether a mother does or does not get preeclampsia is how much unprotected sex she had beforehand. And what we think is going on, so more unprotected sex, lower chance of preeclampsia. What we think is going on is that actually human reproduction, or not reproduction, but human sex is actually in large part training the maternal immune system to go chill when you see these proteins next, because you're going to see them at the maternal placental interface. So please don't attack. And then what you find is surrogate mothers, for example, who take on other people's babies, don't know about mum's proteins, they don't know about dad's proteins in the fetus, and they have the highest rates of preeclampsia, up to 40-50% rates of preeclampsia in that case. So there we think it's just an unfortunate side consequence of a system in which the fetus does have to bury quite deeply into the mother to get all those resources. But if it goes awry and the mother starts attacking it, then you get preeclampsia. The only cure for which, by the way, is emergency delivery.
Well, I mean, you talk also about some of the defense mechanisms that we have to address harmful mutations. I mean, are there differential rates of mutation across different parts of the genome? Are there differential levels of protective mechanisms across different parts of the genome?
To a first approximation, the repair enzymes would just go, you've got a bit of damage in your DNA, let's repair it. They operate the same everywhere. But that being said, there is variation between genes and between genomic regions in the mutation rate. we estimated, for example, and this seems to be true for most species.
So this would be like you want to protect the stuff that's important, and you don't really waste time on the cosmetic stuff, right?
Yes, if you could, that would make perfect sense. But of course, the problem is, can anything actually know what's important enough to repair it? Now, what we do know is, for example, in bacteria, there's a repair system called the MMR, mismatch repair system, and that somehow does focus onto the genes and not the stuff in between the genes. So when you take those away, what you see is that the stuff between the genes keeps the same mutation rate, but the stuff between the genes has a much higher mutation rate. That's because it's focused onto the genes. So yeah, the genes are more important than the non-genic bit. So yes, we see that. There are similar claims that have circulated since the 1970s that it might be the case that we have a lower mutation rate for some classes of genes than for other classes of genes. It's not at all clear whether that is true or not. That's still, I would say, very much up in the air. But what we do know, where we have fairly good evidence, is certain genes will be replicated rather late. So every cell will undergo a replication, then another replication. They copy, copy, copy. And some bits of the DNA will typically copy late on, and some bits of the DNA will typically copy earlier on. And the ones copied earlier on tend to have a lower mutation rate than the ones copied late on. The mechanism is not quite clear, but one thing that does appear to happen is that in yeast also, where we see this, is that we've sort of got two different repair systems. One is slow and careful, and one is fast and panicky. And it looks as though we do slow, careful for the early replicating stuff. And then we go right at the end, you go, oh, right, no, we really need to get this done. And we switch and go to fast, panicky. And we think that's why we might have a higher mutation rate late on. But there's not a lot of evidence that variation in the mutation rate around genomes is itself adaptive, i.e. more important genes have lower mutation rates. There's been one or two claims of that, but they've never really stood up.
Well, of course, you talk about genetic diseases, and you talk about how they're quite prevalent among humans compared to other species. I mean, is that because simply we have these crazy genes, right? These high mutation rates? Is it due in part to the fact that we don't do a good enough job kind of cleaning it up, right?
Well, yeah. So the mutations are stuff that happens that doesn't get repaired. In effect, some of it you can't repair because you can't go. I don't know which is right. So something happens and you've got a choice between, you have a C and a T mismatch, for example. You go, well, shouldn't be like that, but I don't know which was the one I really need and want. So
disease means it's not so bad that it kills you, right? So, right.
There's a whole series of human genetic diseases that have no name. And that's because these are the ones that hit the mutations that hit genes that are so important early in embryo development that if you get it wrong, the embryo simply dies. But the nature of genetic diseases is odd. They're not the most, the genes that cause genetic diseases are not the most important genes. If you have a mutation in the most important genes, you're simply not a baby. You die in utero. And so we only classify genetic diseases for things that have popped out of the womb and now either immediately or subsequently need to go and see a doctor. So they're in this slightly middling zone of I'm important, but I'm not that important. If you were that important and you had a mutation in you, the embryo is dead, and we don't give names to diseases, quotes, genetic diseases that kill early embryos. They're just things that fly under the radar, but they probably explain why we have, in part, why we have this very high miscarriage rate, 15 up to 65%, depending on the mother's age.
Right. And you talk about genetic medicine. I mean, this is an exploding field, right? And so, what can we imagine will be the state of this medicine in the coming years?
I think the answer to that is going to depend very much on where in the planet you're sitting and how wealthy you are. So just to put this into a little bit of context, let's take a relatively straightforward, well understood genetic disease like haemophilia. So haemophilia, sometimes called the royal disease, because Queen Victoria was a carrier of it, and one of her sons actually had it, and it's
Romanov.
also Yes, and also circulated through the Russian royal family through the 19th century as well. What does it do? So haemophilia means if you cut yourself, you don't have the proper system to stop the blood from flowing, i.e. a coagulation system. And so a small cut or a tooth extraction, et cetera, can actually lead to unending bleeding. And in fact, Victoria's son, who died, at the time, the life expectancy for a haemophiliac was 11. Because literally you just sort of fall over, bang your head, you're dead. Normal 11-year-old fall over, bang your head, and you're fine. But you get a bleed on the brain, and it doesn't stop if you're a haemophiliac. And he actually lived to 32, I think, and had a fall and banged his head, uncontrollable bleed. Before that, he was basically wrapped in cotton wool, as you might imagine for a royal. So there we had an understanding that it must be genetic. So the 19th century, they clearly understood this as a genetic disease of some variety. They didn't know quite what it was. And then as you work through the system, they go, all right, we've worked out what this is. It's a genetic disease. And so with that, they go, okay, now we can do some tests to just diagnose. So the first thing you do is, okay, we can diagnose it. And then having diagnosed it and understood it, you can then go, okay, now that we've understood it, we can only both do diagnosis and some sort of therapy. So with the diagnosis, you could go, you just wrap these folks in cotton wool, and they don't bang their heads or anything. With the diagnosis plus treatment, we go, okay, we understand now what this is. This is a genetic problem that you're not making a particular protein that's found in blood that will help your blood from clotting. And so then the treatment was, okay, well, why don't you have a blood transfusion? So haemophiliacs would have routine blood transfusions. That's got problems, and so some people go, it's got problems because, particularly in Britain, actually, there was a big scandal where haemophiliacs would be given blood infected with HIV and hepatitis C and all that sort of stuff. Very, very poor. And so people go, well, maybe we can give you a purified version. We now worked out what the protein is. So not just it's something in blood, but we've worked out what the protein is. So we can manufacture or purify just that protein. And then people go, oh, well, maybe we can do even better. Maybe we can make a synthetic version of it. So in fact, there's a synthetic version coming on the market at the moment. So there you've got a path from a sort of understanding that it's genetic through to a diagnosis, through to doing something about it, to the next generation doing something about it. And this is where it gets interesting, because now we can do something else. All of those other treatments, blood treatment, wrap them in cotton wool, blah, 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 is not a cure. So just wrapping people in cotton wool, please do not go out and play rugby if you've got haemophilia or whatever. Not a cure. Giving people blood transfusions or different versions of proteins is not a cure, because the proteins are unstable. You need to keep taking it, and it is sort of like a plaster over a wound that won't stop bleeding. So where things get quite exciting now is because haemophilia is one of them. We can now do a thing called gene therapy. So with gene therapy, what we're doing is saying, I'm not going to give you the protein which is going to decay and you're going to be back here in 2 weeks for another version of it. I'm going to give you the gene. I'm going to give you the right version of the gene. You can now make this protein in perpetuity. So one treatment, in principle, gives you the gene that you should have, that just about everybody else has. You had a mutant version of it. You can now make your own version of this protein. You're effectively cured, and that is a real cure. So that's gene therapy. Now, I make it sound nice and simple. It's got a number of problems. One is the delivery system, and people can get very bad immune reactions to we put these things in viruses to deliver, because viruses have worked out the trick of getting into cells, but people can respond badly to the viruses. And so one of the early gene therapy trials actually killed the patient because they had a massive immune response to what we call the vector, the viral vector of this. So, the treatment itself can be, in many cases, can be highly invasive and quite difficult. So the gene therapy, for example, for sickle cell disease and thalassemia that now are live in both the US and UK and Europe, basically involves the patient having to have more or less the same as chemotherapy for a blood cancer for about a month. So you take out the blood cells from the bone marrow, but then you have to clean out the cells that are left there. Otherwise, they'd simply outcompete the ones you're about, and that is basically chemotherapy. So it's not it's not a nice treatment. It's not an easy treatment by any means. But the other point is it's not a cheap treatment. So these things are coming in costing millions. And the economics works like, well, it would have cost you an awful lot to have blood transfusion or whatever every 2 weeks for the rest of your life. Actually, one treatment costing about $1 million or $2 million in some cases is actually better off. You're financially better off doing that. So when people are looking at the financial books, so to speak, of should we do gene therapy, they'll look at it and go, actually, it does make sense to put money into that. But then that, of course, does mean it's very much a first world cure. And many of these are actually, particularly things like sickle cell disease and thalassemias, these are very much third world diseases for interesting reasons, but they're not typically high prevalence diseases in US or European populations, but that's where the money sits, as it were. But in the money's worked out such that, so in Britain we have 2 regulatory authorities. One say, is it safe, and do we recommend it? And the others look at it, goes, is it safe? Can we recommend it? And does it financially make sense for our NHS, for our health system to actually pay for it? And actually they've come down, or actually does make sense. It does make financial sense for us as a health system to pay for that. So sickle cell sufferers, for example, in the UK at the moment, they can apply to have gene therapy for sickle cell disease. So yes, there is an interesting new world of genomic medicine just around the corner, as it were. Why do we need it? Because we've got a rotten genetic lot. Yeah. But given that we've got a rotten genetic lot, we've also got big brains, and medicine is anti-evolution in many regards. You know, evolution is why we keep on having these genetic diseases, and medicine goes, well, you might have them, but we're gonna stop them having their effects.
Now, of course, none of this impacts the germline, right? And so,
well,
I know there's been some experiments there that are very controversial. I mean, can you envision a time when we could get in and start tinkering with these mutations and kind of get rid of them for good?
Yep. Oh, great question. Yes. So everything I've told you so far about gene therapy is what we call somatic therapy. So if you've got sickle cell anemia, if you've got hemophilia, we could give you the gene so you don't have hemophilia and you don't have sickle cell anemia, but you do nonetheless have the original mutations in your germline. So your kids could still get hemophilia, they could still get sickle cell, they could still get thalassemia, any of these genetic diseases. So we're not removing...
Well, in fact, one would expect that gene would become more prevalent in the population, right?
Well,
to the extent that we...
Exactly so, because this is effectively what you're doing with all medicine, is every time you keep somebody alive who would have died because of their genetic mutation, you're keeping that mutation in the population where selection would have kicked it out. That's the sense.
We need more medicine. Each generation, we need more medicine.
Yes, yeah. And I think there's an argument that we might be accumulating these things. But yeah, medicine is anti-selection. Selection is kicking these things out of the populations. In human conditions, it's a tragedy. That's a kid dying of cancer. It's a kid dying early of sickle cell anemia or whatever. We can keep them alive, but it keeps the mutation in the population. But what is different, what is potentially different, is to say, okay, we're not only going to treat you, we're actually going to change all of your DNA, so your kids will also not have it. And that's known as germline therapy. And people are very, very hesitant about this. So there was one, Dr. He, H-E, this gets very confusing because I'm going to have to say he said, anyway, but he and he, did actually present evidence of so-called germline therapy. So how you do this, you take out a very early ball of cells, as in an IVF embryo, and you can edit that. And if you edit all the cells in there, and he claimed to do it at the 1-cell stage, it's not clear that all the cells did get modified. Some of them might have been a bit later. In principle, you now have an individual whose genetic problems, as much as you can say it's a problem, have been rectified. And that means that not only is that baby, when it grows up, not going to have the genetic disease, but in turn, their offspring too will not have the genetic disease. Now, this was highly controversial, not least because of the gene edited that he actually wanted to do, he actually did. So it turns out in this case, the father had HIV, and there is a protein called CCR5, which sits on the surface of cells, and there's a mutation that's known that blocks HIV from entering cells. And so the aim of this particular gene therapy was to change the embryos so they had the HIV blocking version of CCR5 because the father had HIV. Now, this is controversial because there's a much simpler cure, much simpler solution to this, which is you actually just have to wash male sperm while doing the IVF, and a sperm wash will prevent them getting HIV. But beyond that, HIV is not now considered a lethal, incurable disorder. The great irony of all this, which is often missed, by the way, is that when he gave this talk at this conference on gene therapy, the very, very next talk up was a talk about the ethics of germline gene therapy and why we shouldn't do it. It must have been a very strange interaction of one person saying, oh, look, I've just done it, and the next person turns up and says, you really shouldn't have done that. But it is seriously considered. Now, the problem is we do not know what harm we are doing. So there are things we call off-target effects. Many of these germline, many gene therapies, also have effects we just simply don't understand. So some of them we insert the gene into your DNA. Now, some of the times we know when that's happened, it's upregulated cancer-causing genes. And so one early gene therapy trial had to be stopped because the gene actually inserted, it turned on the neighboring cancer-causing genes, and 2 of about 12 patients, I think, died of cancer or so. The French who were doing that trial put their hands up straight away, said, we've got a problem, cancel the trial. They actually wrote to the regulator, say, we have to cancel this trial. There's something going on here that we don't understand. So you have that sort of problem with the CCR5 one. This was a CRISPR edit rather than just giving somebody a new version of the gene. This was an edit to the version that they have. This can cause what we call off-target effects. So we don't know it was only that part of the DNA got edited. There could be other parts of the DNA that get edited. We're never quite sure. Also, it's possible that you could actually end up with an individual who is a mosaic, that some of the cells would have the edit and some of the cells wouldn't have the edit. What effect would that have? And if this is such a great mutation, why don't all humans have this great mutation? Maybe there are some knock-on consequences of having this mutation. It does have a big role in the brain functioning, for example. And it turns out that individuals with this actually have a different memory. Their memory isn't the same as normal memory. It's actually a better memory, rather odd, and the same is true in mice. They have a better memory. But whether having a better memory is a good thing, I don't know. So there are all sorts of knock-on consequences. So whatever you do with gene therapy, I think you could make the argument that we are playing with the gene pool. Either you're doing somatic therapy, in which case you're keeping harmful mutations in the population at a higher frequency than they should be, or if you do the germline one, you're absolutely affecting frequencies and mutations in populations because you're directly altering them, but you're also having these other knock-on effects. So I think the consensus at the moment, so there's a moratorium on it. You're not supposed to do germline at all at the moment. But the consensus at the moment, I would say, is that there may be a moral case to be made for some conditions which are currently looking as though there's absolutely no prospect of a cure, but are very severe. So I think people are sort of talking about, for example, there's a very early single locus, single gene, early onset dementia. People say, well, actually, maybe there's a case that if you know you're a carrier of this, maybe there's a case to say edit the embryos there to prevent that sort of thing coming down later in the family history, as it were. But for the most part, I think most people go, look, if there's a viable cure, even if that means a somatic gene therapy, then you go with that. The problem with the brain ones is there's an awful lot of neuronal cells up there that would need editing. And then on top of that, you've got to get things over what we call the blood-brain barrier. So there's a very good barrier between the blood and the brain to stop things in the blood getting into the brain. And so actually just delivering things to the brain is very difficult. This all being said, I think one of the most exciting things that came out a couple of weeks ago was an initial report of some gene therapy for Huntington's disease, which is single locus neurodegenerative disease. Very small trial so far, but with, great prospects. I think 75% showed remarkable slowing of progression of the disease, which I found really surprising. I thought gene therapy for brain conditions was going to be one of the last that we're going to be pick off, as it were. But no, all credit to the bunch in London who've managed that. So that's going on, I'm sure, to a next phase of trialing. But yes, I think there are very profound moral questions about germline editing. And I would say that the genetic community as a whole is, for the most part, as far as I can see, behaving very responsibly, which I'm very happy about.
Well, look, I mean, if we zoom out and look at things in a Darwinian timescale, now that we have a relatively large population, should we expect the mutation rate for humans to go back down to that of other mammals, let's say?
And give it, yes, if we give it long enough, if we can sustain this population for a good old while, then yes, the theory would say it would go back down. However, you do have to think about the timelines of these things, and for a mutation to go from rare to common, so we're thinking, okay, now we could get a bunch of mutations that reduce the mutation rate a little bit. How long is it going to take for them to go from being rare to being common? Now, here you have the double whammy that actually the amount of time it takes is also a function of the population size. Because the longer the population size, right, the more individuals have to be changed, as it were, over time. And we have an estimate that it's something of the order of 4 times the population size. So if our population size went up to, effective population size, about 1 billion, let's say, just for ease of calculation, that would say it'd probably take about 4 billion years. 4 billion generations. With the human generation of about 20 years, that's, 80 billion years? So, yeah. You're right. Come back in 80 billion years, and let's see.
And even if the mutation,
I'm not holding out any hope that it's going to happen any time in the short term.
And even if the mutation rate went down, we would still be stuck with all this clutter, right? That
we'd still be stuck with the clutter. That will take more time. And nearly all of these problems, I think, of reproduction, I don't think that's anything to do with the effective population size. This is reproductive compensation, which would still be there, which is what we think explains our high rate of having too many chromosomes. And maternal fetal conflicts aren't going away anytime soon. And try as we might, we still haven't been able to do anything about preeclampsia.
Well, Laurence, thanks so much. I love the book, The Evolution of Imperfection, right? We're imperfect. We just have to live with it. Thanks so much. Appreciate it.
We do. Thank you. My pleasure.
unSILOed podcast is produced by University FM, elevating the stories of your institution.